All right, let's get one more chapter in before the weekend. This chapter is going to be about antibodies because we talked about binding proteins. We said the antibodies are very tight binding proteins. We're not really going to talk about how antibodies are made, although that's one of my favorite topics. Antibodies are made against a protein you've never seen before, and yet you somehow have a system, your immune system will be able to make a binding protein that will bind something that you've never seen before. How does it do that? Well, it's microevolution, but that's another topic for another day. Once you have the antibodies, how can you measure how well they work, which means how tightly they bind? Now, an antibody is designed to bind an antigen. That's the one piece that you need to know. Antibody is made by you, and it's made to bind the thing that generates antibodies is the antibody generator, or the antigen. Here's an example of, I believe this is hen egg white lysozyme, just a regular old protein that they've built, they've trained, they've given it to some animal, and the animal made antibodies against it. So now we have the antibodies, and they will, you see how they fit together like a big 3D puzzle. The antibody is a really cool structure. What it is it's actually a pretty complex structure, 4 chains and 12 immunoglobulin domains overall. Remember how I mentioned that those were domains used by the immune system? This is the way that they're used. They're used in immunoglobulins, 12 to an immunoglobulin, joined together into 2 chains. The red one is called the light chain. The blue ones are called the heavy chains. So you see that these are basically held together, and you only have two units that are both blue and red, and they have 4 immunoglobulin domains each. These are the 2 arms of the antibody, and it's like at the end of it, you see how on the previous slide, you see the antibody is binding the antigen with the very tip of this, and this is exactly the same coloring as on this one. You have the very tip of the antibody is the binding site. And so you have 2 binding sites, and then at the bottom where it's all blue, that's sort of a handle of the protein. You talk about that a lot more in immunology. It's how the immune system grabs onto the antibody once the antibody has grabbed onto something else. So there's basically 2 ways of talking about an antibody. You can talk about it the biochemistry way, which is green and yellow. You can talk about light chain and heavy chain, number of domains. Or you can talk about the variable region and the constant region, because the bottom of the antibody is the same for all antibodies because it's the way that the body grabs onto it. The top of the antibody is where the antibody changes depending on the antigen. And so what you'll have is you'll have antibodies that have different variable regions depending on which antigen they bind. Because they have 2 binding sites, you get BOGO binding, 2 for the price of 1, buy 1 get 1 free. Once you bind one antigen, you actually have another site open to bind another antigen. A lot of times this can let you bind 2 antigens, or if there's a repeating antigen, like on the surface of a virus, you can bind the virus twice if it's big enough. And once you bind a virus, for example, you see right here you have 4 antibodies have bound this little red hexagon that's a virus. There's other cells of the immune system, such as macrophages. And what they do is, phage means eat. So they phagocytose, or they eat. They basically literally take in the virus. The way that they do that is they grab onto the handle of the antibody and they draw it in. If you look at how viruses enter cells, there's actually lots of variations on this basic idea. But this is the main thing of, if you're looking at a cell, this is a bacterium that's being attacked by the immune system. And we've zoomed in, and the artist David Goodsell, my favorite, has shown these in different colors, coloring different proteins differently. So I want you to take a second and just look at this, zoom in on it if you have to, and realize that the green is the bacterium, the warm colors, and I guess red and blue, so the purplish colors, are the immune system, the human body. And so if you look back and forth at this, where do you see the IGs? Basically look for the Y-shaped structures. What color are they? And if you look around, if you look at the pinkish sort of ones, the pinkish sort of ones have Y shapes. You'll notice there's actually some that are like 5 Y shapes joined together. That's actually a weird type of IG. a weird type of antibody called IgE. But again, we're getting out of the realm. Most antibodies that we're concerned with in this class, at least, are IgG antibodies, the prevalent form in the blood serum. And you can see a lot of those little pink Ys that are located here. Basically, the cell system is really complex. And you can also, if you look at the surface of the bacterium, you can see some of these antibodies bound to the green seaweed, which is actually a glycoprotein, which we will talk more about those later. Or it might be a regular protein. It's a protein, a cell surface molecule. Let's just say that for sure. So there's two techniques in which I want to talk about the way biochemists can use antibodies. There's two techniques that antibodies can be used in. There's a way that you can actually use them in an assay called an ELISA assay. We don't do this in this class, but you may have done it in a biology class at some point. You can also use antibodies to select particular gels out of an immunoblot. Both of those are shown on this. On the left, you have the ELISA assay, and what it really is you stick your whole sample to the bottom of a gel. And even if it's a complex sample, a bottom of a well, I should say, the gel is the next technique. So the sample's at the bottom of the well, shown in the middle of the screen right here. And then what you do is you have an antibody that will bind it. You throw that in, and the antibody binds to the bottom of the cell. Basically, the more antibody binds, the darker the color of the well. But the way you make that trick work is you actually make a secondary antibody that will bind the primary antibody. Remember, they all have a constant region, right? So you make a secondary antibody that binds the primary antibody, and this one is attached to an enzyme that will turn a molecule a certain color in the well. The reason why you have to do that is because it's easy to make a plain antibody. It's hard to make a conjugated antibody. And so what you do is you make a lot of the universal conjugated antibodies, which are shown in green here, that will then allow you to make specific antibodies more cheaply, okay? The red specific antibody is called the primary antibody, and it has an Fc region that is recognized by the secondary antibody. It's a little bit of detail, but you might need to know that because it's actually discussed in the paper we're talking about. The other thing you can do is you can actually use your antibody to, instead of lighting up a well in a 96 well plate, as shown for the ELISA, you also can have an antibody that goes in against your gel. And instead of a well, you can have a gel, and the antibody will bind to a specific band. So if you look at this, there's an SDS page gel with increasing purity as you go along. But is the protein that's at the right size in lane 1 really your protein or not? Well, if you have an antibody that binds it, you actually put that on the gel, and then you make lanes 4, 5, and 6. And the lane will light up where that specific antibody binds that specific protein. And so you can see 4, 5, and 6 are showing us right there, yeah, our protein is still there in lanes 1 and 2 because we see that lighting up in lanes 4 and 5. That's called an immunoblot, and you might see that at some point as well. The main thing, and the reason why I really want to make sure that you go through this lecture this weekend, is because Hsieh et al. Has graphs of antibodies binding. It's how they show that their spikes bind antibodies. And so their response is they're using a technique called surface plasmon resonance, which again, I talk about this in my physical chemistry class, Chem 3410, offered in the spring. You all are welcome to take it. And if you take it, you'll be able to do research on the stuff that you make if you take Biochemistry 2 and the stuff you design at the end of this quarter. That's just a little plug for my class. But we measure antibody binding in physical chemistry using this technique. You don't have to know anything about the technique other than when you have more binding, you see more signal. And so if you inject more antibody and you see more binding, how much binding you see is proportional to the signal. And you can actually do a lot of experiments where you inject different concentrations of antibodies. You see the signal go up. By how much it goes up, you can see how much it binds. Remember that binding is an equilibrium and that we express as the KD, big KD dissociation constant. So from these, you can see the different levels of antibodies that are binding, and you can see that you get dissociation constants that are pretty high. These are two different constructs that Hsieh et al. Showed. They showed the S2P construct and the HexaPro construct. And you see that both of them give nice big red lines that go up, and then they are giving good responses. And so actually you can get the big KD, which is 11 nanomolar for the top example, 13 nanomolar for the bottom example. Remember, that's pretty tight. These antibodies work well. What's cool about surface plasmon resonance, you can also get the kinetic constants. And remember that we said the big K is a ratio of the two little k's. And actually you can see that that works out if you can do the math. You can put it into your calculator with the proper exponents. If you divide the little kd by the little ka, you end up with a 10 to the minus 8th number, which is exactly the big KD that they get there. And so I just want you to say that they give you some of the examples of what they did, but this is the part of the methods that tells you how they got these graphs, and there's a couple questions about those on the study guide. Basically, more antibody, higher concentrations, gave a higher response, which means more binding. And they can put that together into a hyperbola, just like we talked about at the beginning of Chapter 5. So they look at the level of binding, how high does it go, and you can get from that what the hyperbola of binding that goes up as it goes up. You get this out of there. So here's some unpublished data from McLellan that I took, and I believe this is published now because this is about a year old. And they actually took their HexaPro, and instead of just seeing the antibodies bind to it in general, they actually put it into mice. And they saw that HexaPro worked better than S2P for mice. If you look at the HexaPro, it's in red, and the mice, so S2P is on the left, HexaPro is on the right. The HexaPro numbers are higher, and that means tighter binding from antibodies. If you can understand antibody binding, you can actually look at these immunology papers yourself. And here's another thing where they tested therapeutic monoclonal antibodies. This one was called N31. They tested it against HexaPro. And I just want to show you, they're doing a different kind of graph. It's not hyperbolic on the far right, but in the middle you can see that there's antibody binding responses, and they did this by surface plasmon resonance as well. And you see this is even the same person who did our paper. This is Hsieh of Hsieh et al., if you look at the corner. I believe this is published now. Oh yeah, I can zoom in on that, and you can see that they have picomolar binding at this point instead of nanomolar binding. And you can even compare the little k's if you want to, but we're just concerned about the big k's right now. So basically from these are the data that McLellan was using to be able to show that these constructs work well, and they generate antibodies, and they work with the monoclonal antibodies, so they might help to develop new monoclonal therapies. It's all good. And so there's, and just as you might have guessed, because they can get COVID structures, I want to show you just antibody binding to spike, because we can do that with cryo-EM. And if you see remember that the whole thing about the way the spike works, it actually works as a trimer. And if you do cryo-EM of the whole trimer, you see some with the receptor binding domain down and some with one of them up. And I think this is just a really good, clear example of what it looks like when the RBD flips up. And I just want you to look at the antibody. I believe that this is one where the RBD has flipped up to bind the antibody. This is an antibody that binds the receptor binding domain, but it binds it in the up conformation. If the receptor binding domain is down, this antibody will work a little less well, but the good news is it fluctuates back and forth. But you can see the light chain and heavy chain of the antibody. This is just the business end, the variable region, binding to the red receptor binding domain. And you can see that it's sort of, I believe that it binds in the up configuration. So if you can get cryo-EM structures of antibodies that work, we can make a bunch of those, and we can see, well, where are the antibodies binding? And that's where this comes from. This comes from Jesse Bloom's lab, who's actually one of my favorite scientists at the Hutch. I always follow him, and he's actually active on Twitter. So he explains his work on Twitter. So I highly recommend him as a follow, the good side of Twitter. But here's an example of like the receptor binding domain, and they did the surface where they got these different blobs of the spike protein bound to different antibody fragments. And you can see that the different antibody fragments, labeled with these weird labels in different colors, bind at different places to the spike protein. And so you can look all around here and you can see that these look different. These antibodies are binding at different places. Some of them are binding to one side of the RBD, some of them are binding to another side. And basically he plotted this out. A lot of sequence data, a lot of structural data, a lot of cryo-EM. And they found out that different antibodies bound different parts of the receptor binding domain, and these might act differently in neutralizing the virus. Okay, so they have 4 different examples of different ways that antibodies can bind the different places that they can bind the receptor binding domain. And this means that when the spike protein mutates, it's going to affect one of these kinds of antibodies more than others. So how do people make it? Can we tilt the deck? Can we use a certain kind of antibody as therapeutic antibody that's harder for it to evolve away from? And one of the cool things about this paper, one of the reasons I like it, is I took it right here, is that remarkably, when you see the word remarkably, pay attention. Remarkably, combining the escape maps where the antibody mutates with these functional measurements predicts which mutations are selected when the spike-expressing virus is grown in the presence of individual antibodies. In other words, by knowing which antibodies the virus is growing in, we can predict where that virus is going to mutate, because it's going to mutate to get away from those particular antibodies. But one immediate implication of our results, counter to prevailing wisdom, the antibody cocktails do not have to target distinct regions of the RBD in order to resist viral escape. If you just look at that, you can see that there are lots of pairs of antibodies that share no common escape mutations, which means that if you combine them, they will work well together, okay? You don't have to worry too much about the details. Basically, this tells us which antibodies to put together as an antibody cocktail, because we don't want to put 2 antibodies of the same type together, but if we put 2 antibodies of different types together, it's much harder for the virus to escape them. And that's why determining which types of antibodies these are, where they bind, is so helpful and important. Okay. So I just want to remind you, we'll be talking, this is all about antibodies. We're done talking about them. We're going to talk about hemoglobin next, and then we'll be ready for the next test. So Monday, we'll be talking about hemoglobin. I want you to look at lecture 11 and do the foundations exercise that is assigned on Achieve for you. Myoglobin is actually a hyperbola that binds the same way as what we already showed in class, but hemoglobin's binding curve is actually S-shaped. In Foundations, they show you that curve and they have you work through it. So work through as Foundations says for the assigned problems. If you do that before next lecture, you will be ready to talk about hemoglobin, and that has the implications of sickle cell disease. So that's enough for today. That's more than enough, but we've got lots of relevant stuff for you to learn. And I will see you on Monday, and we'll be ready for the test in a week.